You know, there is an entire universe unfolding all around us, right under our noses. It's this bustling, invisible world of life that has a huge impact on our health, our food, everything. So today, we're going to pull back the curtain and see exactly how scientists make this microscopic world visible.
I mean, think about it. This was the massive challenge that completely stumped early scientists. It's one thing to say, hey, microbes exist. It's a whole other ballgame to actually isolate one specific type from this complex soup of life and study it. How do you possibly grab just one suspect from a crowd you literally cannot see?
Okay, so before we dive in, let's get a few key terms down. The very first step is taking your sample of microbes, that's called the inoculum, and introducing it to some kind of food source.
Now, when those microbes take hold and start multiplying, that beautiful growth you see, that's what we call a culture.
And the whole shebang, the entire intricate process of growing and nurturing these microbes in a lab, that's cultivation. It sounds simple, right? But trust me, it is an incredibly powerful science.
But it wasn't always this precise. Oh no. The early days were a total mess, a scientific quagmire, really. And the solution, the thing that broke the whole field wide open, didn't come from a famous scientist's brilliant insight. It came from a suggestion, straight out of the kitchen.
So to really get this, you gotta picture how things used to be. You had giants like Louis Pasteur using liquid broths, but just imagine trying to pick out one specific microbe when they're all swimming around in a murky soup. It's pretty much impossible. Robert Koch knew he needed a solid surface. He tried potato slices, too inconsistent. Then he used gelatin, which was a step up, but it had a couple of huge fatal flaws. And that's when, in 1882, the real breakthrough happened. Fanny Hess, the wife of one of Koch's lab assistants, just casually suggested, hey, why not try agar? It's something she used to make jellies.
So what was the big deal about agar? Why was it so revolutionary? Well, it all came down to two simple things. First, unlike gelatin, most bacteria can't eat it, so your nice solid surface actually stays solid. But here's the real game changer, the melting point. Gelatin turns back into liquid mush above 28 degrees Celsius. That's way too cool to grow the germs that make us sick, which love our body temperature of 37 degrees. Agar, on the other hand, stays rock solid all the way up to 100 degrees. That one little difference, it changed microbiology forever.
So, we've got our solidifying agent, our plate, so to speak. But what do we feed our microscopic guests? You can't just give them all the same thing. You've got to think of culture media as a precisely crafted menu, tailored to the incredibly diverse and often very, very picky dietary needs of the microbial world.
And this menu has a lot of options. You've got your enriched media for the gourmet eaters, the fussy ones that need extra stuff like blood or egg. Selective media are like a bouncer at a club door. They have ingredients that stop unwanted microbes from growing. Differential media are super clever. They have color-changing indicators that react to what the microbes are doing, giving you these awesome visual clues. Enrichment media are liquid broths that give a specific microbe you're looking for a head start. And transport media, they're like a little stasis pod, keeping microbes alive but dormant on their way to the lab.
Let's zoom in on a classic, blood agar. This is a perfect example of a medium that does double duty. It's enriched, because the blood gives extra nutrients to those picky bacteria. But it's also differential. See, some bacteria make enzymes that literally shred red blood cells. That's called hemolysis. This creates a clear zone around the colony, which we call beta hemolysis, a huge red flag for pathogens like Streptococcus pyogenes. Others just cause a greenish bruising, or alpha hemolysis, while some don't do anything at all. It's a critical visual clue right there on the plate.
Okay, so we've designed the perfect menu. Now, how do we actually serve it? We're moving from the what to the how. And let me tell you, plating isn't just slopping bacteria onto a dish. It's a skillful craft, and it's absolutely essential for reaching the holy grail of microbiology, a pure culture.
The undisputed workhorse of every microlab is the streak culture. The goal here is beautiful in its simplicity, progressive dilution. You start by smearing your sample in one small corner. Then you sterilize your little wire loop, cool it, drag it through that first streak just once to pick up way fewer bacteria, and spread them into a second area. You repeat that a couple more times. By the time you get to that last section, you've thinned out the microbes so much that single individual cells land all by themselves. And each of those single cells grows into a perfect isolated colony.
Now, what if you want the exact opposite of isolated colonies? Well, that's where a lawn culture comes in.
Here, the goal is to create a solid, uniform carpet of bacterial growth across the entire plate. This is absolutely critical for things like antibiotic sensitivity testing. You lay down these little paper discs with different antibiotics on the lawn, and then you can clearly measure the kill zones around them to see which drugs are most effective.
So far, we've been talking about growing microbes that are totally fine with the air we breathe. But what about the ones for whom oxygen isn't life-giving, but is actually a deadly poison? Yeah, cultivating anaerobes presents a whole new set of challenges. So why is oxygen so deadly for them? It really comes down to biochemistry. When you use oxygen, your body creates toxic byproducts, like hydrogen peroxide. Now, we have protective enzymes like catalase that act like a cleanup crew to neutralize these toxins. Obligate anaerobes, they don't have that cleanup crew. So when they're exposed to oxygen, these toxins build up and literally poison them from the inside out.
To grow them, we basically have to create a completely oxygen-free world for them. The classic way is the Macintosh Fields jar, which uses a catalyst to chemically scrub the oxygen out of a sealed container. A more modern take is the gas pack system, a simple disposable pouch that does the same thing. We can even use special broths with ingredients that chemically absorb any dissolved oxygen. But the gold standard? That's the anaerobic cabinet, a big sealed glove box that lets a scientist work with the cultures without ever exposing them to a single molecule of oxygen.
Okay, we've just gone through some pretty detailed, nitty-gritty lab techniques, but why does all this meticulous work really matter? Let's connect this foundational science back to its vital, real-world impact.
See, mastering microbial cultivation is absolutely essential for modern medicine and biology, and it all really boils down to these 7 critical pillars.
At its very core, cultivation lets us isolate the bad guys from an infection. It helps us tell the real pathogen apart from all the harmless microbes just hanging around. This lets us identify the culprit and figure out what makes it tick. And maybe, most importantly for a patient, it lets us test which antibiotics will actually kill the bug, which is crucial for guiding treatment. Beyond that, it's how we get the raw material for diagnostic tests and vaccines, and how we keep living libraries of microbes for future research.
And you know, this isn't just about medicine. These exact same principles, creating the right menu, the right environment, and carefully isolating what grows, are what we use to search for life in the most extreme places on Earth, from fiery deep-sea vents to volcanic lakes. And that work is the foundation for astrobiology and our search for life on other worlds, like Mars.
Which brings us to this final, absolutely fascinating thought. For all of our incredible techniques, for all our clever media and fancy cabinets, scientists estimate we still can't grow about 99% of the microbes that exist out there. Their needs are a complete mystery to us. So as we look to the future, the biggest question isn't just about what we found. It's about what incredible discoveries, new antibiotics, new technologies, maybe even entirely new forms of life are still waiting for us in that vast, invisible, unculturable world.